Today's presentation will be about how ergot and head blight influences barley and their link to mycotoxin production and potential ways to control infection and mycotoxin production via manipulating the environment. Ergot is a fungal disease that comes from the claviceps species that infects cereal crops. It infects the crops just before the flowering period of barley crops. This infection produces its most visible symptom, that the blackened grains it produces, known as sclerotia. It is one of the oldest known fungal diseases that goes back as far as 600 BC on an Assyrian tablet, where it was described as noxious pustule in the ears of grains, in reference to how deadly it was to those who consumed it. Head blight is a fungal disease caused by Fusarium species. The symptoms for grains are observed on the head and neck of the grains and causes bleaching. Within barley, the disease does not spread anywhere beyond this. During humid conditions, orange spots can also form on the spikelet. Head blight disease grows most effectively in wet and warm weather during the maturation and growth periods of the cereal crops. The reason why barley is the crop of interest for the study is that it's an economically significant crop that represents a sixth of the world's total global grain production. It's used in many different products, including but not limited to beer, whiskey, animal feed, and bread. In Scotland, in 2018, the grain sold for a total of £3.18 billion, and its whiskey production brought about an revenue of £5.5 billion. The reason why ergot and head blight are a concern for barley production is they produce deadly substances for human consumption known as mycotoxins. They are so deadly that even having a 1% of infected grains present within the barley shipment would mean that that shipment will be discarded. In addition to this, the diseases causes harm to the crop. For example, head blight causes necrosis of the barley cell walls in order for improved ease of digestion, and can also, if present in the soil, cause similar damage to the roots, which reduces the crop's capacity to absorb nutrition. In ergot, the damage is less pronounced, but infected grains are inedible due to the danger of consumption. They also, post-infection, can risk infecting second rounds of the crops in the soil if ideal agronomic practices, such as crop rotation to a non-cereal crop and tillage, if it is not applied to the soil. Ergot infection begins when ascospores from the wind land onto at-risk wild and forage grasses and crops such as barley in the spring. Next, the stigmas of these plants trap ascospores, which leads to infection. These can infect the ovaries of crops within 24 hours of exposure. During exposure, the ascospores develop hyphae which colonize the ovary and will grow into the ovary axis, henceforth mimicking pollination. Next, the infected ovary grows a stroma that produces honeydew, which attracts pollinators to the crop, which further increases the spread of infection. Other ways this infected honeydew can spread to other plants is through rain, farm equipment, crop head contact, or splashing to nearby crops. This process continues until the sclerotia matures within 4 to 5 weeks, replacing the seed for a black cereal head fungi. Barley has an increased chance of infection through a secondary inoculum as opposed to infection caused from nearby wild grass fields. In addition, infection occurs just before flowing, as during flowering there is an improved resistance of ergot in the crop. Therefore, conditions that delay pollination, such as cool, wet weather, increases ergot spread. During infection, a sclerotium is formed, which is a white-like mycelial tissue that contains dark pigmented storage cells for the fungi's energy, and the sclerotium, that is the black outer shell that protects the fungi from UV damage and desiccation, as the inner body is susceptible to both forms of damage. During harvest, the sclerotia is harvested with the grain. However, some fall into the soil, which makes the next batch of grain at risk of infection. It takes 25 days in the soil for ergot to hit the next stage of its life cycle, within temperatures of 0 to 10 degrees Celsius, for vernalization to occur. This transforms the sclerotium into stalk-like mushrooms known as the stromata, which is where sexual dimorphism starts, which is the stage of the ergot's life cycle where sexual reproduction starts. When the bodies are ready to burst, the fruiting bodies form perithecium, a fruiting tube-like structure where the spores produced by sexual reproduction is discharged. When discharged, the ascospores will try and reinfect cereal crops and wild grasses, therefore restarting the infection cycle anew. Head blight infection starts when spores of Fusarium species and on barley crops. The crops are most susceptible from the early flowering stage. The second most vulnerable stage of infection is the soft dough stage, where developing grain kernels are still soft. This results in the formation of tombstones, and mycotoxin production will occur. These spores can also infect the roots through the soil, or with seeds that have been infected with head blight. This will damage the crop throughout its development cycle, therefore reducing productivity more so than infection of the grains alone can do. This is caused by a reduction of nutrient uptake. Early infection of the crop also increases its susceptibility to disease. Also, there is increased uptake of mycotoxins from the soil from head blight contaminated soil vs uncontaminated soil, which will increase the likelihood of the grains being rejected when being selected for further production. Mycotoxins are secondary metabolites that are produced by fungi. These are of interest for research because they pose risk of harming animals, insects, other fungi, plants, and fish. Side effects of consumption of mycotoxins typically include mutagenic, teratogenic, and estrogenic-like effects. Ergot produces a group of mycotoxins known as ergot alkaloids. The most known ergot alkaloids are ergometrin, ergotamine, ergocristine, ergocryptine, ergocine, ergocornine, ergotamine, egocristin, ergocryptinine, ergochroninine, and ergocinine. These mycotoxins are harmful to both humans and livestock. In humans, they causes gangrenous-like effects caused by vasoconstriction of the blood vessels, leading to restriction of supply of blood to the limbs, causing the loss of limbs and in some cases death. It additionally causes nausea, hallucinations, alterations in endocrine functionality and effects on the cardiovascular system. Three main groups of mycotoxins make up head blight mycotoxins. These are the deoxynivalenol, nivanols, and the 4-acetyl nivalols. These toxins are harmful to plant and animal alike, and the quantity produced is dependent on the host crop. These mycotoxins causes rapid tissue necrosis of the infected crop. Furthermore, the severity of the damage caused by it is dependent on the amount of deoxynivalenol enzymes the fungi produces to degrade the cell wall. The fungi also utilize callus, chitonases, and xylonases to further break down the crop for consumption. For humans and livestock, the side effects of consuming from head blight infected crops. The side effects depend on the dosage and duration that the dose. This can result in metabolism disorders affecting the breakdown of proteins, fats, and carbohydrates, which can lead to issues synthesizing nucleic acids, thereby causing liver and kidney failure along with increased risk of cancer and death. For the ecosystem, whilst head blight exclusively infects grain crops, the mycotoxins can pose potential damages to the wider ecosystem. This is because of potential runoffs into nearby water samples. However, it needs the presence of other toxins such as herbicides and pesticides to also run off to pose a significant risk. In the case that it does, it will aid in the destruction of species diversity within freshwater ecosystems and any species that may need to drink from the river, leading to a potential risk of bioaccumulation of carnivorous species. Mycotoxins provide a risk to production post-harvest. Firstly, ergot is too difficult to separate due to similar masses, which is why it's usually discarded at 1% levels within the grains. Additionally, infection can spread if the grains are not properly dried during storage through head blight. Another difficulty regarding processing is that during the washing and grinding phases, the mycotoxins present will spread throughout the entirety of the product. Furthermore, even if the heads of the crop are not directly infected, it's possible to absorb mycotoxins through the soil, which is why crops do need to be tested for mycotoxins. Some processes can be used to reduce the potential damage caused by mycotoxins, such as using UV light to damage the toxins into less harmful forms before washing. However, the decontaminated toxins still pose a health risk. Not much is known directly to the link to mycotoxin production to environmental conditions. However, it is known how the environment affects the life cycle of head blight and ergot. They both prefer wet and windy environments when in the air, so the spores can easily infect the head and spread across the crop rather than it be localized in a singular area. Furthermore, the temperature conditions needed for ergot to grow in the soil is understood to grow from 0 to 10 degrees Celsius in order to hit the sexual reproductive stage. However, the proliferation within the soil can be prevented using tillage, but causes long-term damage to the soil and its biome, so alternative measures of infection control within the soil needs to be considered. So my current plans for my project is set into two objectives. The first goal of my PhD is to measure the production of mycotoxins and see how this level of production differs in accordance to changes in the environment. This aim will be achieved via growth experiments within an environmental chamber, where light, temperature, and humidity levels will be controlled, and will compare the differences in infection rates and production of toxins between a wild type and agricultural variety of winter wheat. This will be achieved using bioassays as a measuring tool on levels of mycotoxin production. The second goal of my project is to be able to translate this knowledge into an agricultural setting, so I will be looking how to tweak an agricultural system to have close to as possible replicated environmental conditions as seen in the chamber. I can see this being achievable through mulching and irrigation methods being the primary two methods that can be tweaked to achieve this goal. However, other ideas may crop up later in my literature research.